One of the major challenges in molecular biology and drug design is understanding how proteins interact, especially when we don't have a reliable 3D structure. In many cases, researchers are left with only the amino acid sequence, while the exact spatial conformation is missing. This makes it extremely difficult to predict binding regions, for example, in protein-protein or antibody-antigen interactions. Conventional docking tools depend entirely on high-resolution 3D models. Without those, they simply can't function, leaving a major gap in early-stage discovery, especially for unstable or flexible proteins. At Burnham Labs, we've developed a breakthrough method, linear docking. It analyzes interaction potential directly from the amino acid sequence, no 3D structure needed.
We present the developed algorithm for a step-by-step transparent and understandable search for interaction areas, each of which will be characterized by numerical physical quantities. As you know, there are a huge number of programs for docking proteins, mimics, and other molecules. The main difficulties are initially correct task of contacting surfaces, so that the search does not turn into an astronomical explosion of all possible options. Control of the correctness of the received docking data. To solve these problems, we propose the following methodology. 1. Use information about the structure of protein-forming domains, alpha-helix, beta-sheet, disordered structure. 2. Use the linear docking method developed by us, which allows us to determine the most stable binding regions according to the amino acid sequence. We propose to carry out the check with entire domains, and to divide the molecule into structural elements to determine the most contact amino acid sequences by the method of linear docking. The interaction between sites will be characterized by numerical indicators, which will allow for visual gradation. 3. Return to the 3D structure of the participating compounds and designate the found domains that are subject to further docking and verification. 4. Specify contact surfaces in molecular docking programs. 5. Step-by-step verification of the 3D PDB file structure.
Let's move on to a full description of the calculation part with examples. Next, we will introduce you to two developed methods, Algorithm 1 and Algorithm 2, using the example of searching for interacting domains of the Nap1 and Mdm2 proteins. Let's start with a description of the features of the domain structure of these two proteins. Mathematical algorithms have been developed that allow determining the detection of proteins' active regions and detecting the stability of different regions of protein complexes. Linear docking, by analyzing the potential energy matrix of pairwise electrostatic interaction between different sites of the biological complex, such as the homodimer of the histone chaperone Nap1 and the homodimer Mdm2 which are responsible for the entry of a whole protein molecule into biochemical reactions. Human murine Mdm2, Mdm2, is a 491 amino acid long phosphoprotein. In the central part of the protein there are many acidic regions. The C-tail contains a zinc-binding domain. This part of the protein interacts with a variety of regulatory factors as well as multiple ribosomal or nucleolar proteins. The C-terminus also contains a ring domain that has been shown to be responsible for the E3 ubiquitin ligase activity, as well as the binding of the Mdm2 and Mdm2. Next, we provide a step-by-step calculation for determining the contacting domains. The diagrams show the structure of the Mdm2 protein, both in three-dimensional representation and in linear representation. Particular attention will be paid to the search for the homodimerization domain.
Let us consider in more detail the structure of the protein of the histone chaperone Nap1. The structure of the protein Nap1 can be divided into several sections: unstructured N and C tails, domain 1 and domain 2. Consider in more detail domain 1, which is responsible for the dimerization of Nap1. During the dimerization process, an interaction occurs between the longer two helices of two proteins in opposite directions. The dimer is further stabilized by the A2A3 loop, the A3 helix, and the A3A4 loop that wraps around the base of the A2 helix of the dimerization partner. In figure, a schematic representation of the domain 1 and domain 2 structures is presented. The following figure shows a scheme of the formation of the Nap1 homodimer by two proteins of the histone chaperone. In blue color, which is denoted by domain, one of the first histone chaperone, Nap1 with indication of a 2 and a 3 helices, which take part in homodimerization. Orange color represents domain 1 of the second protein of the histone chaperone, Nap1, and also shows the A2 and A3 helices. In monochrome, the rest of the proteins that are not actively involved in the formation of the Nap1 homodimer are shown.
One of the main rules for verifying research work, both experimental and calculated results, is the agreement of the results obtained by different methods. Therefore, our calculated results are supposed to be obtained using different calculation methods to confirm the correctness of the results obtained. Let's start with a description of algorithm 1. This algorithm has been developed to search for protein sites responsible for protein interactions. We write the amino acid sequence of the protein under study in line as vector 1. Below, we write a shorter sequence that corresponds to a certain domain of the protein, be it an alpha-helix or a disordered region, and we also write the second vector in a line. The shorter amino acid sequence of one-dimensional array P2 moves along the longer amino acid sequence of one-dimensional array of the protein P1 with some step. In our example, the step is equal to 1 amino acid. In each step occurs the formation of a short section of the amino acid sequence of protein P1, equal to the length of the shorter one-dimensional array of protein P2. Each new segment of the one-dimensional array P1 corresponds to the length of the polypeptide chain on dimensional array P2. 5 pairs of one-dimensional arrays P1 and P2 were successfully formed when the one-dimensional array was shifted by one amino acid residue along the one-dimensional array P2. After finding all the participating pairs of vectors, we build a matrix of potential energy electrostatic interaction between their amino acid residues. These matrices will have a square form. Further, from each of these matrices we calculate the value Leconte and construct a graph of the dependence of Leconte on the order number of the amino acid residue of one-dimensional array P2, where Leconte is the condition number. Thus, each resulting value of Leconte will correspond to a strictly defined segment of one-dimensional array P2. Note that on the graph it is postponed opposite the first amino acid residue of a segments P21, P22, P23, P24 or P25 one-dimensional array P2. Figure shows graph dependence of values Leconte on the sequence number of the polypeptide chains of participating one-dimensional arrays 1 and P2. In this example, as we see from graph, the smallest value of Leconte corresponds to the interaction of the vectors P13 and P23. The dotted line indicates the amino acid sequence of one-dimensional array P2, which participates in the formation of a biological complex with a one-dimensional array P1. The value of the value Leconte is placed opposite the first amino acid of the remainder of the segment of the one-dimensional array P2. Let's move on to consider the second algorithm. Algorithm 2. We developed the second algorithm for detecting interacting regions of protein molecules. The scheme search interacting sections is shown in the figure. In this algorithm, we take whole amino acid sequences of the two proteins P1 and P2. For selecting interacting sites, we shift the frame of a specific size along two one-dimensional arrays of proteins P1 and P2. Note that algorithm 2 can be used to analyze the interaction of two proteins which have identical sites of interaction during the formation of a dimer. After finding all the participating pairs of one-dimensional arrays, we build a matrix of potential energy electrostatic interactions. The matrices will have a square form. Further, for all matrices we calculate the values Leconte and construct a graph of the dependence of Leconte on the order number of the amino acids, residue of the participating one-dimensional arrays.
In this section, the interactions of the short amino acid sequences Mdm2 and the long sequence protein have been modeled. Numerical simulation was performed according to the developed algorithm 1 and 2. Thus, the purpose of this section is to test the developed algorithm 1 and 2 in determining the most active interaction regions of a full-length protein with a short polypeptide sequence. At the same time, they tried authors to choose a common scale for presenting the obtained graphic data for all the calculations performed in order to facilitate understanding and to allow the reader to visually compare the results of the obtained data. In addition to the graphical representation, we also gave 10 minimum values of low COMDA, with a list of the corresponding interacting amino acid sequences. Numerical calculation of the interaction of Mdm2 436 to 482 and Mdm2. Let's look at the calculation results obtained for the Mdm2 dimer. A search was carried out for the polypeptide chain Mdm2, which is most inclined to form a complex with the Mdm2 protein ring domain. To achieve this goal, we took a Mdm2 protein domain polypeptide region 436 to 482 and numerically calculated its interaction with the Mdm2 protein according to algorithm 1 developed earlier. In this case, the short sequence of the protein Mdm2 436 to 482 shifted along the long sequence of the protein Mdm2 at intervals of one amino acid residue. As a result, for each pair of the obtained one-dimensional arrays, a matrix of potential energy of electrostatic interaction was formed, and the value of log COMDA was calculated. The value of log COMDA was plotted opposite the first amino acid residue section of the Mdm2 protein upon interaction with Mdm2 436 to 482. The results are shown in figures. As we can see, the set of minimum values form a cluster from the C-terminus of the Mdm2 protein. Thus, it can be concluded that the sequence Mdm2 436 to 482 plays an active role in the dimerization of the Mdm2 protein and is most likely to form stable biological complexes in the C-terminus region of the Mdm2 protein. Next, we analyze the numerical calculation of the homotomer formation Mdm2 according to algorithm 2. We use the amino acid sequences of the proteins Mdm2 and Mdm2 and formed the corresponding one-dimensional arrays by shifting the frame along two polypeptide chains at intervals of one amino acid residue in order to identify the most active regions of the interaction of the proteins studied. The 10 minimum values of log COMDA for each calculation obtained are tabulated in the corresponding graph. We now turn to the analysis of numerical calculations of the interaction of 2 and Mdm2 proteins at a frameshift length equal to 20 amino acids, as well as 30, 40, and 50 amino acid residues. 10 minimum values of log COMDA are presented on the charts. As can be seen from the figures, the greatest number of minimum values lies at the C-terminus of the Mdm2 protein.
The final calculation section is devoted to the obtained data, which allowed us to determine the homodimerization region of the Nap1 protein. The presented results clearly demonstrate that breaking down the amino acid sequence into structures allows us to obtain self-consistent results more quickly and accurately. Let's start with the results obtained using algorithm 1. In this case, the amino acid sequence was cut out from the 81st amino acid residue to the 150th amino acid residue, which is an alpha-helix in the Nap1 protein. Then this section of the polypeptide chain was presented as a short vector, which was shifted along the entire amino acid sequence of the Nap1 protein. The results for each such shift were presented as a matrix of potential energy of electrostatic interaction and subsequent calculation of the condition number. The 10 minimum values of the condition number are shown in red on the graph. Let's move on to the results obtained using algorithm 2. The figures show the results for a shift frame size of 50, 60, and 70 amino acid residues. This shift frame size is close to the size of the alpha-2 helix. A shift frame with a size of less than 40 amino acid residues leads to chaotic results. The graph shows 10 minimum values of the logarithm of the condition number. As can be seen from the graphs, we observe a qualitative comparison of the obtained results. Use the linear docking method developed by us, which allows us to determine the most stable binding regions according to the amino acid sequence. We propose to carry out the check with entire domains, i.e., to divide the molecule into structural elements, to determine the most contact amino acid sequences by the method of linear docking. The interaction between sites will be characterized by numerical indicators, which will allow for a visual gradation. Parsing the structure into domains. 1. Alpha spirals, beta sheets, turns to intramolecular bonds that provide the formation of domains. Conducting calculations on domain interactions. Checking the correctness of the 3D model based on the available data. 1. Obtaining the physical characteristics of interacting molecules. Stability, entropy change, Gibbs energy, constant dissociation, enthalpy change, the thermal dissociation, potential energy of electrostatic interaction between all amino acid residues taken in pairs.